My name is Tareque Hassan Mukut. I'm a first year graduate student at the Department of Chemistry and Biochemistry, and currently I'm working in Dr. Bunamo's lab. The topic I'm going to present is bio-inspired deuteranic materials for bioremediation and biomedical application. The phosphoethanolamine moiety introduces deuteranic characteristics to a molecule at physiological pace. Cellulose modified with phosphoethanolamine can be found as a key component of robust biofilm of Escherichia coli because gram-negative bacteria added significantly to the modified polymer. In the top right corner of my poster, you can see the biofilm life cycle, how bacterial biofilm forms through the attachment of gram-negative bacteria to the surface, irreversible attachment, maturation, and dispersion. The formation of biofilms and subsequent encashment of bacterial cells in a complex matrix can enhance the resistance to the antimicrobial agents, which making this organism difficult to eradicate and control. We propose to create a novel biomimetic materials with deuteranic properties using polyvinyl alcohol in place of cellulose. It is anticipated that the resulting PE-PVA material will attract the gram-negative bacteria to edit the polymer surface similar to the PE cellulose. We chose multiple synthetic pathways to synthesize our target materials, and I mentioned two synthetic schemes in my poster, which I am currently working. We had some synthetic challenges because of the solubility of our starting materials, but finally we overcome it. In our preliminary research, we synthesized the novel PEPV material through the formation of PO bond on the polymeric materials. We used FTIR, phosphorus NMR, and proton NMR to characterize the molecule, but further characterization is required to determine the physiochemical properties. Still, our initial results are quite promising. I attach the spectrum in the middle of my poster, the FTIR and the NMR spectrum. And you can see the transparent gel substance on the top of the NMR spectrum, which is the product we synthesize. Now I want to mention the future perspective of my material we developed. As I mentioned, it is a novel material, so we have to characterize its physical properties and chemical properties properly. We want to use it as a bioremediation tool to remove the bacteria from polluted environment, especially from soil and water. We want to incorporate bactericidal characteristics by adding membrane-busting peptide to the amine group of this material. We will assess the possibilities to make wound healing hydrogels. We can also use this material as a biosensor to detect the gram-negative bacteria from polluted environments. And we can also detect the gram-negative bacteria from the patient sample by using this material. And surgical switcher is another target we want to work. Finally, I would like to thank you all for showing interest in my poster and thank you for your time.